Guinhita Bintang. You can call me Fortran. And today I'm going to teach you all about biology for SAT subject test, and I'll be covering 6 chapters. This is our first chapter, Chapter 1, Cells Biology. So today in this lecture, I'm going to explain and go through the molecules in the cells first. So what is molecules? Molecules is a really small particle, but it do have a smaller particle. And whenever we talk about molecules, we're going to separate it in 2 parts: water and biomolecules. Biomolecules are molecules that is in biology, which is really small: carbohydrate, protein, lipids, and nucleic acid. So these 4, we're going to go through all of it. Now our first one, what are molecules? First of all, I'm going to go through what we call atom first. Atom is a very small particle that atoms and atom compose together to give molecules. So whenever we talk about atomic structure, we have to think of 3 things: proton, neutron, and electron. Proton, this one, has a charge of positive, and electron, this one, has a charge of negative, which is actually really opposite, while neutron is at the middle, so it's 0. It doesn't have any charges. The relative mass, what I'm talking to is proton and neutron. These 2 things combined together give atomic mass, which is really heavy, while electron has less density and less mass. We say it's 0. The location of proton is in the nucleus. Neutron is in the nucleus. As I mentioned, it composes together. An electron, it stays at the electron shield, just like its name. Now, where is the electron shield? We have to have a look of this thing, atomic structure. This part right here at the middle composed of 2 things, proton and neutron. And that 2 things, again, make up atomic structure. And whenever we talk about, atomic mass. And whenever we talk about atomic mass, we're not going to say anything about electron, because electron has a really light weight comparing to proton and neutron. Now, if you look at the electron can skip from place to place. So electron can be transferred. And we call this thing is that electron can be excited. And this is really important. Electron can be excited. So from this, I'm going to put it in red. What do I mean by excited? Well, it's not going to just stay in its atomic structure, but instead it will go through other, and this lead to ETC, electron chain, electron transport chain. And this will always leave what we call ATP, or adenosine triphosphate. And we're going to talk about all of this through again when we talk about cellular respiration. There we go. And that's the whole thing you need to know about atomic thing. Now, atoms and atoms together need to have a bond. And the bond is something that connects the atom together to give a molecules. So what are bonds? How can they attract? Okay, so whenever we talk about attraction, in chemistry or biology, we have to think of bondings. There are 2 types of bond, mainly: chemical bonds and intermolecular bonds. Chemical bonds are divided into 2 things: ionic bond, which I will say give and receive. One of it will show positive ions, and one of it will show negative ion. We'll go through it deeper. Covalent bond is about sharing electron. So I'm not going to give you, you're not going to receive mine, but we will share it together. And in another word of bondings, we also have an intermolecular bonds. Intermolecular bonds have polar attraction, hydrogen bonds, and non-polar attraction, which this one is actually really weak. Now we're going to go through the chemical bonds. First one, ionic bond. As I say, ionic bond, one of it will stay in positive ions, while another one stays in negative ions. Why? As I say, electrons, if it's in the normal part, it will be like positive, 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 negative, negative, negative. What I mean is positive and negative have to be equally in order for them to state. But if I say this one has one extra one and it just wants to give it away, so what can we do? So another one has an extra, well, another one wants it. What we have to do is take that positive and give it away. So this one have more negative and it showed negative, while the one that received it will show positive because it has more protons coming. Proton, the positive, electron, the negative. Just to remind you again. Okay, so as you see, one extra, give it away. Another one you have to know is what we call covalence bond. Covalence bond is to share it between atoms. So it can be divided into two parts, non-polar one and a polar one. The non-polar one will share it equally. As I mentioned, it's a non-polar. I'm not going to have more En than you, which I mean electronegativity. So they will share it equally and form two identical atoms. While another one is polar, they will share it unequally. So if they share it unequally, this depends on En, electronegativity. And this thing will share it and form two different atomic structure. So these things are actually like water, as water is H2O. So it's shared like this. Some kind of that. So now that's all for the chemical bonds. We're going to go through the intermolecular bonds. Intermolecular bond, it's divided into three things: polar attraction, hydrogen bonding, and non-polar attraction. The polar attraction is when we talk about things like hydrocolloids or this thing. It depends on positive and negative En. So this thing is actually when two or more atoms form a bond, making the entire molecules either polar or non-polar. So we're going to note that either polar or non-polar. So this is what we call dipole to dipole attraction. Well, hydrogen bonds. Hydrogen bond, or another word we call it H bonds. This will given some kind of polars and polar. So I'm going to note it there. It is connect between polar groups. So polar and polar connected together form hydrogen bond. But not just polar. It has to be hydrogen connect to a En. Again, electronegativity. What are those? Well, it is fluorine, oxygen, and nitrogen. How do I know? Well, it depends on the periodic table, which some kind look like this, and fluorine has the most En. So anything come closer to En, which I mean fluorine, it will have a higher En. But just to know, F is the highest. Now let's go to our nonpolar attraction. Nonpolar attraction is an attraction between nonpolar and a nonpolar. So it is actually really weak attraction. And in other words, we call this attraction van der Waals or London dispersion force, which is the same thing as van der Waals. Yes, so that's all. Now we're going to have a little bit look at our cutie note that we will have two different vocabulary, one called phobics and another called philic. We'll see these kind of vocabulary again when we talk about the cell membrane thing, but I'm just going to say this here first. Phobic, bigs is like bored. So it doesn't like B and board. It doesn't like water, so it does not dissolve in water. Doesn't dissolve in water. While the philic, which L stands for like, this is a way for me to remember. So this means it dissolves in water. Or in another word, hydrophobic is also known as does not. This means non-polar. While hydrophilics mean it's like and it dissolves. So this means it is apolar. So some kind of example for non-polar are like lipids and all of those oily things. But polar are just anything that dissolves in water. So here goes. Water. Now water we are going to talk about has a property. 5 different property that we have to remember in order to do SAT. So first property is water is cohesive and adhesive. What do I mean by cohesive and adhesive? I'm going to go through right here really quick. That you have to know the vocabulary of cohesion and adhesion. What do I mean by cohesion and adhesion? Cohesion is when water molecules stick to water molecules, while adhesion is when water molecules sticks to other molecules. And this is actually really important to what we called transpirational pull. And if you're thinking, what is transpirational pull? Don't worry, I'll go through it. So let's say we got a tree, and this tree is like 10 meters high, which is really high comparing to us. We, a small human, standing right here, water our plants. Do you think that the water that goes through up here is only from the root pulling it up, or just pushing it up? Actually, no. From the root, it will get into one of the point, and then that's it, because it doesn't have that much force. So that much force that leads the water up here is to pull, and that pulling is called transpirational pull. And that transpirational pull have to work for cohesion and adhesion, working together, just like climbing. There we go. Another property that we have to know is water is a universal solvent. Whenever we talk about solvent, we have to think about 2 vocabulary, solute and solvent. What do I mean? Actually, I have to say this one is solution. So whenever we talk about solution, we have to think that solute dissolve in solvent. For example, I got a beaker. That beaker is filled with water. Oopsie. So if I got a salt and water, and if I put these 2 things together in a beaker, I will see that the salt dissolves in water. So my solvent is water and solute is my salt. That's it. Now, if I talk about the next property, I'm getting stuck with the mathematics. So our third property is about water regulates the temperature. What do I mean? Well, imagine you're in a pool, swimming pool, and it's a hot sunny day that it's about 38 degrees Celsius, which is really hot. But as long as you jump into the water, which is full in here with the pools, you'll feel eventually it's cold. Why? Because this property, that it regulates the temperature and it doesn't evaporate that easy. Just like if you are swimming here in the ocean, floating there. Even the weather is really hot, but still you'll feel cooler if you're down here. So this is another property. Next property is that the water expands as it freezes. So if we put a bottle of water into a freezer, we will get the ice, which has intentionally, like if this is a bottle. So if our water is right here, the level of like the ice might be like here freezing out here. Why? Because it expands. And this is caused why ice float. Why does ice float? Because it has lesser density. Less density. Why less density? Because expands area. Yeah. And our last property of water in SAT is that water is and biochemicals are connected. How? See this thing? Are you familiar with it? Yeah, this is called photosynthesis. While this one is actually what we call aerobic respiration. And we'll go through it deeper when we talk about it. There we go. And these two things, just so you know, are waste product. Remember this thing though, SAT have the question like this a lot of time. Now, that's it for the first thing, which is like our first step. We get through the molecules, we get through the water. Now, the next one is our biomolecules. As I mentioned, biomolecules are divided into four things: carbohydrate, protein, lipids, and nucleic acid. I'm going to go ahead and write it briefly. Carbohydrate, protein, lipid, and our last thing is nucleic acid. So just so you know, before getting into, it has things to do with the chemistry things. So our carbohydrate will consist of carbon, hydrogen, and oxygen. Protein will contains carbon, hydrogen, oxygen, nitrogen. Well, lipids is same as carbohydrate, carbon, hydrogen, oxygen. And nucleic acid is carbon, hydrogen, oxygen, nitrogen, and phosphorus. Now we're going to go through all of it. Our first one is called carbohydrate. Carbohydrate can be divided into three things. First one, we call it monosaccharide. Monosaccharide is the thing I mentioned, carbon, hydrogen, and oxygen composing together and form three things: glucose, fructose, and galactose. It actually forms a lot more, like deoxyribose we found in DNA, or ribose we found it in RNA, and that's it. So these things connect together, as mono means one and di means two. So mono connected together give disaccharide, which has glucose, sucrose, fructose. Whenever we talk about glucose, we have to know that glucose plus glucose form maltose. And as we see here, sucrose is from glucose plus fructose. So sucrose, I'm going to write it here. That's number one, that's number two. Glucose plus fructose form sucrose. And our third one is lactose, is from glucose plus our last one, galactose. So I misspelled things here. Galactose. Okay. And as it forms more and more, this will give you polysaccharide, which poly means many. And what are examples of polysaccharide is like starch, glycogen, cellulose. So starch is stored in plant. These are just some facts you might need to know. Cellulose stores in plants, like in the cell wall, and glycogen stores in animals. Chitin is actually stored in fungi. Yeah, and these are like polymers of glucose. See? Glucose has a shape like this. So if I go ahead and copy, or actually just move, you see that more glucose, more glucose, more glucose. Like if you zoom in, we'll see that glucose, 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 and all of these things connecting together form polysaccharide. Now that's it for carbohydrate. We'll go ahead and go to our protein. Protein, what SAT is about the structures and also enzyme. So our first one is about the structure. The structure of protein, first one is called primary. Primary is an amino acid sequence. Secondary is helical form in protein, result from H bond. And H bond is what we talk about when hydrogen is connected to high EN, FON, which is fluorine, oxygen, and nitrogen. Tritary, the third one, is when like the secondary together form, and the last one is like the whole thing that's formed together. So have a look at the picture here. These whole thing like a chain thing, this is the primary protein. So primary protein is about amino acid, and amino acid is actually like the smallest part whenever we take like proteins in. So if we eat protein, it's like meat, it will get digest into amino acid. So this amino acid together form primary together form this thing, which this thing is called secondary. A function depends on the structure, and the structure is formed in tritary one. So the third one here form a function, and these are like paddle sheets and those things. And the last one is quaternary, which is the one that is ready to be functioned in order to let our body move around and do those things around. And I'm going to go through this deeper in a chapter of humans. So here it goes. Another fact that you might need to know in order to do SAT is that, H bond are found in secondary, tertiary, and quaternary. So these things can actually be denatured or it could be destroyed by high temperature. While the primary one, which doesn't have an H bond, the one that makes the primary has a wrong or like destroyed the amino acid sequence will only be about the mutation. Nothing else could destroy it. pH, temperature, only mutation. So I'm going to have that highlighted. Oops, that's too dark. Mutation only. Here goes. Now, enzyme is like a larger part of protein. So whenever we talk about enzyme, is that it is a large functional protein. An enzyme is not degradable and reusable, as mentioned here. Not. It speeds up the reaction, and the reaction is a reaction to speed the energy of activation, which we call it EA. And it has a graph like this. See? Without enzyme, you have to take that high mountain up here, but with an enzyme, it reduced the EA. So you only have to take the steps. Easier. So it speeds up. Now, whenever we talk about enzyme, there are 2 points. Lock and key models and induced fit. This is just some kind of cute code. You are my substrate to my enzyme, and nothing could ever denature us. Well, let's go through the first one. The induced fit is a models that are used into today that it will induce a little bit, slightly change to let the enzyme take into place with the substrate, while the lock and key model is actually the one that it has to be exactly the same in order for it to just go like, oops, we're made up together. See? Okay, and the induced fit model, as I mentioned, it will slightly change itself for the enzyme and substrate to connect together, and once everything is finished, enzyme will stay the same. So enzyme again is not degradable, and just have happened to be in SAT for many of times. Okay, so this one is cute, so I brought it up here. It is called a competitive inhibitor, which means that substrate and enzyme can't be connected together because there is some kind of weirdos, which I mean like other substance that comes up here, and then it doesn't make enzymes and the substrate fix together. And once again, If enzyme and substrate is complex and together within it, just like this, once it's out, it gives the products and the enzyme is back again. So here goes. Lipids. Wow. The lipids is all about fatty things. So I'm going to divide lipids into fatty acid and glyceride. Fatty acid I divide into saturated and unsaturated. I'm going to write it over here. The first one we are going to talk about is called saturated. The saturated fat is not kind of a good fats, just like it came from animals. It is a solid in room temperature, which is 25 degrees Celsius. It causes heart disease. And this one is very important that you must know, is that saturated fats has only single bonds between carbon atoms. Going to talk about it just in a minute. I'm going to talk through about the unsaturated first. And unsaturated fats, it's more friendly to our body. It doesn't cause us a heart disease because it is from plants. This thing will be liquid in temperature room, 25 degrees Celsius. It is a good dietary fat that I mentioned, and it has at least one double bonds between carbon. What I mean is this two thing is definitely completely opposite. Okay, so example of this is butter. An example of unsaturated is healthy fat. Like this thing actually is really in a solid. Whenever you see fats from a cute little piggy, it is solid. Well, the oily that we cook is actually from the plants, so it's in a liquid. Now, how do we know? It will be about the structure. Like if this and this connect together, C and C, which is carbon, this is called saturated because it only has a single bond. While the unsaturated is like this, it has double bonds, or even at least one double bond. Like if it's like this, it means it can be connects more. So if connects more, it means that the carbon doesn't have only one hands, which I mean bonds. So this is an unsaturated fat. Now we're going to go through the glycerol. Glycerol will be in two parts: neutral glyceride and phosphoglyceride. While the lipids has these kind of things, but this thing isn't really in SAT. So I'm going to go through it really fast, that I'm going to tell you other facts about it. I'm not going to write it, but I'm going to say it. So lipid function, the energy storage is 1 gram gives 9 calories. The biological structure in the main component, it is in the cell membrane, which is like mostly fluid acid. I'm going to talk through it about it. Biology function will be about hormones and steroid. So if SAT tends to ask you what kind of biochemicals is about steroid, it is lipids. Okay? Now go through the nucleic acid. Now nucleic acid is actually really close to this. That if we take, I'm not going to say it, it's too dangerous. DNA and RNA, it's in us, DNA. So this is all about this picture. The RNA usually have like one strand, so it's like that. The DNA, it's deoxynucleic acid. It's in a double helix, so it's look like that. Okay. There are purine. I'm going to note it right here. There are two types: purine and pyrimine. So purine, P-U-R-I-N-E. It is purine. So it is A-G, A and G, purine, A and G. And this has two circular. Well, another one is pyrimidine. Pyrimidine is about C, T, and U, which does only form one. So these are like the thing I mentioned, cytosine, the C thing. G is guanine, A is adenine, U is uracil. Cytosine, guanine, the same things here. Thymines and adenine. So the difference between these two is the T and the U. If you notice here, the T and the U. T for thymine, U for uracil. So whenever we talk about DNA, it's T. If we talk about RNA, it's about the U. Whenever we are doing the transcription and translation things, this thing will really matter. This is just a picture to show you like how they connect. A and T connect together, C and G connect together. You see that bonds right here, this bond here. These are all hydrogen bonds, guys. And remember again, hydrogen bond can be destroyed by high temperature, right? And this is actually about the next chapter we're going to talk about too. And that is all. The next lecture we're going to talk about the cell structure. And that's it for today. Thank you. Bye bye.